Now let us get introduced to antibacterial drugs. Antibacterial drugs is the right term, whereas it can be also called as antibiotics. And it is not a right term when it is called as in whole, because antibiotics are drugs which are chemical substances which are derived from microorganisms, or basically living beings. Now let us go into the classification of antibacterial drugs. They can be divided into 5 categories. They are cell wall synthesis inhibitors, protein synthesis inhibitors, drugs which act on cell membrane, which acts on the metabolism, which is antifolate, and inhibitor of DNA gyrase. So, cell wall synthesis inhibitors, let us have a look at its structure. They are basically penicillins and others. So, they have got a beta lactam ring with thiazolin ring. Let us have a look at its structure now. This is the beta lactam and this is the thiazolin ring. So this is got with the structure, and now let us have a look whether it is cidal or static. So first, looking at these terms, bactericidal and bacteriostatic. Cidal means to kill, so kills bacteria. Static means it inhibits replication or multiplication in other terms. So, cell wall synthesis inhibitors, what it basically does is, this is a bacteria, and let us imagine it has a cell membrane and a cell wall. Now, while it is replicating into two, it again generates a cell membrane and a cell wall. But what these cell wall synthesis inhibitors do is, they make the cell wall go away, and now we have got only the cell membrane, and this one, it is not being able to replicate and it will not survive, because when there is no cell wall, what happens? All water and everything from the environment enters into it, which is hypotonic. Now entering into it, it will eventually swell and burst, then die. So this is a bactericidal drug, whereas protein synthesis inhibitors just inhibit the replication, so they are bacteriostatic. Now, drugs which act on cell membrane, when cell membrane does not exist, this is the cell membrane and this is the cell wall. When cell membrane itself does not exist, it can never survive. So this is bactericidal. Antifolate is bacteriostatic because it acts on its metabolism, and DNA gyrase is bactericidal because it does not allow for the replication. How it happens is that we will see the mechanism of action now. This is just a simple mechanism of action. So there is an absence of cell wall. There is no cell wall, as already explained. There is no cell wall. So water and solutes enter. Water and solutes enter. As they enter, there is a lysis of cell. And the last one, DNA gyrase inhibitors. What they do is they are needed for replication. The DNA gyrase is needed for replication. DNA gyrase is needed for replication, and if it is inhibited, and DNA gyrase, when it gets inhibited, the DNA will break, so no replication will happen. And let us have a look at whether it is applicable for replicating bacteria or not. The first two drugs act on replicating bacteria. Now let us have a look at the drugs which are included under these categories. Cell wall synthesis inhibitors include beta-lactam, glycopeptide, cycloserine, bacitracin, and phosphomycin. These beta-lactams include four under them: penicillin, cephalosporin, carbapenem, monobactam. Now glycopeptide has vancomycin, cycloserine, bacitracin, and phosphomycin are not frequently used. Now protein synthesis inhibitors include tetracycline, macrolides, clindamycin. These macrolides include drugs such as erythromycin and azithromycin. Clindamycin, then linezolid, streptogramin, and aminoglycosides. Here there is one exception. All of these are bacteriostatic except for aminoglycosides, which is bactericidal. An example for aminoglycosides is gentamicin. Now, drugs which act on cell membrane are polymyxins. And a lipopeptide which is daptomycin. Now antifolate drugs include sulfonamides and cotrimoxazole. In cotrimoxazole, These we call as bacteriostatic except for cotrimoxazole, which is bactericidal. And DNA gyrase inhibitors, example is fluoroquinolones. They end with floxacin. They are ciprofloxacin and norfloxacin. With this, we complete the classification and a few extra other points. We did not check out on what spectrum it acts. Penicillin. Penicillin acts on gram positive. Cephalosporins act on gram negative. These have both positive and negative spectrum. Monobactums are negative and rare. Glycopeptides act on resistant gram negative. That is, we give penicillins, we give cephalosporins, and if they do not act on all these, we give vancomycin, which is for resistant ones. With this, the classification gets over, and we are going to look at the decision making on which antibiotic we are supposed to give. Which antibiotic we are supposed to give is decided by the fact that we must have to kill the bacteria. So bactericidal is preferred more than static, because for static we need the immunity to kill the bacteria, to kill the actual bacteria. It just inhibits the replication. And beta lactam, beta lactams, they are less toxic and are more preferred. And if the benefit is more than the risk, it is used. And we are not supposed to combine uh, these kinds of cell wall synthesis inhibitors with protein synthesis inhibitors. That is, cidal drugs are not supposed to be combined with static drugs, mainly because of the fact that cell wall synthesis inhibitors inhibit the cell wall synthesis, whereas protein synthesis inhibitors inhibits the replication. When this inhibits the replication, and this acts mainly on replicating bacteria, the whole purpose is in question. So it must not be combined used. Now, the drugs chosen are on the following basis. If the organism has a cell wall, if cell wall is present, we go for beta lactams because they are more safer. And for gram positive ones and gram negative ones, for gram positive ones we give penicillin, for gram negative ones we give cephalosporin. Or we can also give penicillin, a few act on them, and also carbapenem. And if there is showing of resistance, we give vancomycin. If they are resistant, we give vancomycin. Along with this, we usually give add-on drugs. Add-on drugs are usually aminoglycosides. They are given for severe infection. Example is gentamicin. They are given for both gram positive and negative bacteria. Now, this is the case when cell wall is present. When cell wall is absent, we will go to the condition where cell wall is absent. Now, when cell wall is absent, in for example in organisms like mycoplasma, legionella, chlamydia, for these kinds of organisms, the drugs used are protein synthesis inhibitors. Because it does not have a cell wall, we are going for protein synthesis inhibitors. The examples are protein synthesis inhibitors are tetracycline, macrolides, and clindamycin. These are first line. Then the rest are linezolid and streptomycin. These have increased toxicity, so second or third line drugs. And when the infection is mainly in urinary tract, so we must use the drugs which increase concentration in the urine, or else they are mainly excreted by the kidneys. And the examples for such drugs are fluoroquinolones. They are the first line drugs, but the only unpreferred fact is that it is toxic, but it must be used based on what? It must be used based on the benefit risk ratio. If the benefits are more, we are supposed to use them. Fluoroquinolones are the drugs which end with fluoxetine, ciprofloxacin, and norfloxacin, norfloxacin. Then sulfonamides. The other drugs are sulfonamides and cotrimoxazole. The thing is that these are more of anti-protozoal drugs than antibacterial drugs. So rarely they are used for bacteria. And the thing with aminoglycosides is that they are nephrotoxic, and that is supposed to be noted. The thing with aminoglycosides is that they are nephrotoxic, and the fact is supposed to be noted. And if the infection, now we have seen the infection if it is in UTI. If the infection is in GIT, we give drugs which increase the concentration in stool. Drugs with increased concentration in stool. And it is nothing but fluoroquinolone. Fluoroquinolone, it is the first line drug. And last line drugs are the drugs which have increased toxicity. For example, polymyxins. Polymyxins are for resistant gram negatives. And lipopeptide, daptomycin, only drug. It is for resistant gram positive. Resistant gram positive. And there are a few drug specific details. Which are for peptides. Which drugs are peptides? Glycopeptides and lipopeptides. Only two. Glycopeptides and lipopeptides. They are vancomycin and daptomycin. These only we have seen. What are these peptides? That is proteins. Since they are proteins, they have increased size. So since there is an increased size, there is poor oral absorption. There is poor oral absorption because of the increased size, and it cannot enter gram negative cell walls. Gram negative cell walls have only got aquaporins, which are nothing but water channels. They are very small, and they cannot pass through the increased sized proteins. Hence, the spectrum of these antimicrobials are only gram positive. Only gram positive organisms like Staphylococcus and Enterococcus. Then aminoglycosides. We move to aminoglycosides. This is the first one. Then we move to aminoglycosides. Aminoglycosides have got poor oral absorption. And it is used in monotherapy and polytherapy. Monotherapy and polytherapy. When used in monotherapy, it is only for gram negative aerobes. For polytherapy, we can use it against both gram positive and negative. Now, in gram negative aerobes, what are they? E. coli, Klebsiella, and gonorrhea. E. coli, Klebsiella, and gonorrhea. Now, what are they? E. coli, Klebsiella, and gonorrhea. Why they are active only against gram-negative aerobes? Because simply these aminoglycosides do not enter into the cell. First of all, to enter the cell, such as a gram-negative bacteria, it has to get an action potential, and with that action potential, it tends to pull in the aminoglycoside. So this action potential is generated, mainly transmembrane potential is generated by ATP used with oxygen. It gives transmembrane potential. This transmembrane potential allows the drug to enter. And since the oxygen use must be there, it is working only against gram negative aerobes. Whereas in polytherapy, we have got when it is used with beta lactams, mainly these beta lactams, no, they will break down the cell wall. So again, when it is used with aminoglycosides, it only has cell membrane to enter. So it easily enters the cell, and now it has got what kind of effect? Bactericidal effect. It can kill. So bactericidal effect it has got. With this, the antibacterial drugs introduction gets completed. This is for taking screenshot. Thank you.